We're now in the fourth part of our virology lecture. And we're now going to start talking about the RNA viruses. We've completed the DNA viruses, we're moving into RNA viruses, and there are more RNA viruses than DNA viruses. So for our RNA viruses, we have enveloped, we have the non-enveloped, we have in our non-enveloped single-stranded genomes and double-stranded genomes. So here is a nice little schematic of all the different RNA viruses. And again, we're going to focus on those that cause human disease, and we're starting out with the double-stranded RNA viruses. And we're going to start out by talking about the reoviruses. And one of the notorious reoviruses is a virus called rotavirus. Now, rotavirus is a segmented double-stranded RNA virus. It's transmitted fecal-orally. It survives very well on inanimate objects. It causes gastroenteritis much more commonly in infants and children 6 months of age to 2 years of age. So this is a test that I used to do when I worked in hematology. We did a lot of these tests, and we would classically get these very yellowy, watery diarrhea poops from 1 and 2-year-old kids. And back then we did these tests in hematology, and we did not have a hood, a class 2 hood in hematology. So what we would do is we would open up these rotavirus poops from these little babies. They stunk so bad they would stink up the entire lab, and we'd actually burn incense while we were opening these poops and doing the test just to try to get the smell away. Nowadays, you'd likely not open poops out on the benchtop. You'd open them up under the hood. It's not quite as smelly. But that's one of the problems with microbiology. It is a stinky area of the laboratory. So we used to do a little card test. It was a very simple test where you would put the poop in this, it's almost like a pregnancy test. You put the poop in, you'd put a couple of drops of reagent in, and it would turn a color if it was positive. So that's a very simple, quick test. You could do ELISA tests, you could do latex agglutination. Now, epidemiologically, the winter to spring months are going to more commonly have rotavirus, but you can get rotavirus infections any time of the year, but it is more common in, the kind of the spring-like temperatures, and it is a common nosocomial infection. So I kind of have a funny, I think, rotavirus. I'm not sure what virus, but I was visiting a friend of mine, and when I was flying down there in Florida, she warned me that all of her kids had just had this terrible intestinal infection. She also had just gotten it, and so did her husband. So basically, the entire family got it from the kids. I was not coming down there for a couple of days. I was stopping off. I was going to a conference first. And so she said, it should be fine by the time you get here. I've cleaned the entire house. I've sprayed Lysol on every surface. And I got there fine, great, first day, go here, go to the zoo, go there. The kids are all better. Everybody's better. And the night before I'm leaving, I was so deathly ill. I really wanted someone to it was awful. This gastroenteritis was awful. So it was either rotavirus, because it started in the kids and rotavirus is so common in kids, or it was a norovirus, which we will get to in a few slides. So we're going to move to the single-stranded RNA viruses. Not many double-stranded RNA viruses that cause human disease, but now we'll go to single-stranded. And we'll start out with the arenaviruses. And arenaviruses can cause a variety of diseases. You can get lymphocytic choriomeningitis, which is similar to an aseptic meningitis, a viral meningitis. So we talked about meningitis when we were talking about bacteria, and we have bacterial meningitis or meningitis, and then viral meningitis tends to be called aseptic meningitis, and that's because when someone is suspected of having meningitis, you do a spinal tap. You are immediately going to do a lot of tests on that spinal fluid, one of which is going to be a culture in a petri dish to see if you get bacteria to grow up. Well, classically, in someone that has viral meningitis, you don't get any organisms growing up on your appropriate agar petri dish, which is why it's called aseptic. Lassa fever virus is one of the arenaviruses. So classically, this virus is transmitted like our Lassa fever virus, which causes a hemorrhagic fever, not a very pleasant disease, is through inhalation of rodent urine. So rodents get infected and they can excrete the virus in their urine and feces, and then humans come into contact with that and end up getting infected. So arenaviruses can be diagnosed serologically and through nucleic acid-based tests. Bunyavirus can include, there's a lot of different viruses in these major divisions. So the Bunyaviridae are what's called arboviruses, and arboviruses are commonly transmitted through an arthropod vector. So a mosquito, tick, sandfly, and we've already talked about bacteria that are transmitted through your arthropod vectors, and this is a virus or multiple viruses. So the viruses that are included here are lacrosse virus and Crimean Congo hemorrhagic fever virus, or CCHF. Also, Hantavirus is in this whole family too. Now, unlike lacrosse and Crimean Congo hemorrhagic fever, which are transmitted through an arthropod or insect, hantavirus is transmitted through rodent excretia. So hantavirus was identified because a lot of individuals all of a sudden came down with this terrible viral infection. Several people died, and it was in the Four Corners region of the United States. I believe this was back in the 80s. I'm not sure the exact date, but it wasn't that long ago. And they identified this virus, hantavirus, and they found that all of these individuals in this area lived in homes that were infested with rodents, and they had a lot of dust. So when they would sweep up the dust that had the rodent excretia in it, the dust would come up into the air and they breathed it in, and that's how a lot of them got infected with it. So the diseases that are caused by these viruses are encephalitis, pneumonia, or hemorrhagic fever. Diagnosis, usually serological diagnosis through antibodies detected in spinal fluid. Calicivirus. Now here we go with our noroviruses, which is what I might have had. It was either a norovirus or rotavirus when I visited my friend in Florida, who is a clean freak to begin with, and when she told me she scrubbed the entire house and sprayed every surface down with Lysol, and yet I still got it, just goes to show how incredibly transmissible these viruses are. So fecal oral transmission, disease, and I could account for this if this is what I had, is nausea, vomiting, and diarrhea. Awful. and it can lead to hepatitis or liver disease in especially in pregnant women, which can be a highly fatal infection. So noroviruses and Norwalk virus is a type of norovirus, can be diagnosed through electron microscopy, PCR, EIA, as well as serology. However, I will say most people get nausea, vomiting, and diarrhea for a day or two, feel awful. I mean, really awful. But then they're fine. So most people actually don't ever submit any sample to get diagnosed. Usually they're diagnosed based on symptoms. So noroviruses, there's been some outbreaks recently on the news where cruise ships, they get norovirus on them, and half of the people on the cruise will come down with this terrible nausea, vomiting, diarrhea, and they can't get rid of the norovirus. They'll scrub the ship, not allow any cruises to happen for a while, scrub it, then finally say, okay, everything's fine, let's start a cruise again. And again, half the people, there's been some cruises where every single person on board got sick. So very, very tough to get rid of and highly transmissible. Coronaviridae family, or your coronaviruses, are transmitted. There's various ways: direct contact, aerosol transmission. The exact transmission of some of the viruses is not completely known, but coronaviruses cause a number of diseases. So common cold, gastroenteritis, and coronavirus is also the cause of SARS, or severe acute respiratory syndrome. So SARS made headlines several years back where a lot of people came down with this terrible respiratory tract infection. A number of people died, and they linked SARS to being a coronavirus. So diagnosis can be electron microscopy. A lot of times it's serology, and you can of course use nucleic acid testing. And when there happens to be an outbreak happening, it's diagnosed a lot of times just based on symptoms. The filoviruses or filoviruses are two more biothreat agents are included in this category of virus, and that is Ebola virus and Marburg virus. So transmission is thought to be through infected monkeys, as well as person to person through body fluids. So Ebola and Marburg viruses cause the hemorrhagic fevers. So basically with these viruses, your organs just bleed out. So these are more highly fatal infections. Mortality can be up to 90%, and individuals that work on these viruses have to work on them in a biosafety level 4 facility where you are in a suit and hooked up to an airline inside a complete full air pressured suit. So diagnosis can be electron microscopy, and you can do cell culture in monkey kidney cells. The Flaviviruses family include arboviruses like the yellow fever virus, dengue virus, West Nile virus, the Japanese encephalitis virus, and the St. Louis encephalitis virus. So the flaviviruses are transmitted through arthropod vectors or insects, usually the mosquito, not all of them, but most of these are the mosquito. And diseases are encephalitis, the St. Louis and West Nile viruses cause encephalitis, dengue fever, and yellow fever. So diagnosis of these are serologically as well as antibodies in spinal fluid. So more serological based diagnosis, as well as exposure when there happens to be an outbreak happening. You can, if someone happened to be in an area that has the mosquito vector and it's a certain time of year, like summer months where there's a lot of mosquitoes, or someone was in an endemic area, you would suspect some of these types of viruses. Orthomyxoviruses include your influenza viruses. So there are three major antigenic types of influenza: influenza A, B, and C. And types A and B are what cause almost all of the human disease. So, influenza A is transmitted through respiratory secretions and causes the disease influenza or the flu, where you'll commonly get a headache and feel very tired and muscle aches and have respiratory symptoms, coughing, and it could also cause pneumonia, bronchitis, croup, which is that coughing, and ear infections. Influenza A can be detected through ELISA-based testing, fluorescent antibody staining, and cell culture. And epidemiologically, with influenza A, you see antigenic drift that occurs with this virus and antigenic shift that occurs. So it's important to know these two terms. Antigenic drift is a minor change in the antigenic structure of this virus. And what happens is, as the virus replicates, you'll commonly get errors that occur or little mutations that may not cause any problem, but these mutations can accumulate and change the antigenic structure of the virus so that the body doesn't recognize it quite as easily. So antigenic drift can cause outbreaks to occur every couple of years as the influenza A virus accumulates these mutations and changes enough where your body doesn't quite recognize it anymore. And that's why scientists try to figure out which antigens are on the virus are going to appear and change the vaccine each year. Now, antigenic shift is a drastic change in the surface antigens, and this is caused by adaptive mutation. The virus just adapts to an environment and decides to just completely mutate and change its surface antigens to survive, or genetic reassortment, where you have a virus from one animal that combines with a virus from another animal, reassorts, and becomes a brand new virus that is completely novel to the body, to the immune system, and that's when you have an awful, terrible outbreak, like the swine flu outbreak that occurred, where you will have, initially, you'll see a fair amount of death. Now, influenza B is a milder influenza, again, respiratory secretion transmission, and the same type of detection. Now, in order to detect influenza virus doesn't lyse infected cells. So you can do a heme adsorption assay. So what happens is cells that are infected with influenza virus have hemagglutinins. So if you have your cell culture with your influenza virus infected cells, you can add red blood cells into the culture media, and the red blood cells will adsorb to the outer membrane of cells that are infected, and you'll be able to see it under the microscope. So that's one way you can detect influenza virus. But more commonly, it's going to be a serological based test or just mainly on symptoms, what time of year it is. So influenza is going to be more common in the winter months, and you could really tell by the symptoms that a person would get. Now, the paramyxoviruses are viruses such as measles, mumps, parainfluenza virus, RSV or respiratory syncytial virus, and human metapneumovirus. So we'll start out by talking about measles. Transmission is through respiratory secretions. It's very, very contagious. There is a vaccine. The MMR is a measles, mumps, and rubella vaccine, 3 different viruses. So the measles virus causes measles, can also cause subacute sclerosing panencephalitis, which is a much more severe complication and detected through cell culture and serology. Measles is more commonly diagnosed, especially before vaccine, when kids commonly were getting measles. You can see the little the spots all over the body, the little red characteristic spots. Mumps virus is transmitted person to person in respiratory droplets, and it causes the disease mumps. And mumps is an inflammation of the salivary glands. So what happens is the neck of an infected individual gets very, very swollen. This is one of the classic childhood diseases that all kids used to get before there was a vaccine. It's detected through cell culture and serology, but again, it's more commonly diagnosed through the huge neck that someone comes in with, and there is a vaccine. So you don't see mumps very often anymore. Parainfluenza virus, another respiratory secretion virus. In adults, it can cause upper respiratory infection symptoms, rarely pneumonia. And in children, again, respiratory symptoms, including bronchitis, pneumonia, and croup, which is this awful coughing where almost sort of similar to whooping cough, but a little bit different. So detection, cell culture, shell vial, and fluorescent antibody. And there are 4 serotypes. You can get disease all year around with these 4 different serotypes. Now, RSV, or respiratory syncytial virus, is person to person or respiratory transmission. The disease primarily occurs in infants and children, causing a respiratory type of infection, bronchitis, pneumonia, coughing. It can be grown in cell culture in Hep2 cells. You can use ELISA testing, and you can use fluorescent antibody staining. So the disease is more commonly found in the late fall through the early spring, and you can see nosocomial infections all year round. Human metapneumovirus causes symptoms very similar to respiratory syncytial virus, influenza, and parainfluenza virus, and it's more common in the winter. Now we're going to talk about a different group of organisms, and these are the Picornaviruses, and these include enterovirus, hepatovirus, which includes the virus hepatitis A, rhinovirus, and parechovirus. Now, the enteroviruses. This is a large group of viruses that includes poliovirus, Coxsackie virus, echovirus, enterovirus. Transmission is fecal or oral, and it causes a variety of diseases. So poliovirus causes the disease polio. Polio used to be a more common disease many, many, many years ago before there was a vaccine. However, the vaccine got rid of all of the polio, and so you don't see polio very often anymore in the United States. You can see meningitis, you can see hand, foot, and mouth disease, you can see pericarditis. So the Coxsackie group of viruses can cause all of those. Enteroviruses usually will cause an intestinal type of infection and symptomology, so vomiting, diarrhea, and detection is cell culture, PCR, and serology. Now, rhinoviruses, rhino, nose viruses, these are what cause the common cold. Coronaviruses can also cause the common cold. There are at least 100 serotypes of rhinoviruses. Transmission is respiratory secretion. So we already said the common cold is the disease, and detection can be cell culture. However, it's usually not necessary when someone's sneezing, coughing, runny nose, and you pretty much know it is a rhinovirus. So it's not commonly diagnosed in the laboratory. We're now going to move on to the fifth part of this lecture.